Welcome to another lecture in the series for animal cell culture techniques and application, and this is the third lecture in the series. So, before we proceed any further, let us recapitulate as to what we have learnt so far. So, we learnt that primary cells can be obtained from an organ from mice or from humans, and these cells can be grown in culture under requisite conditions of temperature, pH, humidity, percentage carbon dioxide and so on. Now, if you take these primary cells and then you split them and subculture them in different plates or flasks, then what you are doing is you are creating a secondary culture or you are creating a cell line. Now, these cell lines may have indefinite or infinite replicative potential. Such is the case if you are taking an organ or if you are taking cells from a cancerous tissue, or if you have infected the cells with a virus, then the cells would have an indefinite or an infinite replicative potential. Then you are giving rise to a continuous cell line. But the cells of our body have a finite replicative capacity, which means that they would grow under normal conditions for a certain period of time, after which they will stop dividing. They might be metabolically active still, but they would stop dividing, and then in the end they will die. So, these cells have a finite replicative potential, and therefore, these are the finite cell lines. Now, what are the components that we need to keep our cells happy? So, as you can see here, the first component is definitely the temperature. So, human body has a temperature, normal temperature of about 37 degrees centigrade, and therefore, the cells are maintained at that temperature. So, the whole thing is maintained in something called an incubator, which is a chamber that will maintain under aseptic conditions these cells at a definite temperature, at a definite percentage of carbon dioxide, and under humidified conditions. So, the cells are grown in a medium with amino acids, glucose, lipids, vitamins, inorganic salts, trace elements. We also need to strictly maintain the pH of the media. The pH of the media is roughly about 7.4, and it is maintained with the help of sodium bicarbonate that is present in the media and the carbon dioxide that is circulated through the incubator. So, the bicarbonate carbon dioxide buffering system actually helps to maintain this pH. And as additional additives, we add serum, which provides essential growth factors, hormones, lipids, amino acids, and we also add antibiotics like penicillin and streptomycin, so that it is only the cells that we want to grow that are growing in culture, and the bacteria which may grow, because you have bacteria in the air and that may contaminate your sample. You do not want them to grow because it is a very rich nutrient source. The media is very rich in nutrients and therefore is also ideal for bacterial growth. You do not want the bacteria to grow, so you add antibiotics. So, after this recapitulation, let us go into some other details about how you can manipulate cells. So, this is very pertinent to understanding animal cell culture, because you just do not want to culture cells, you also want to be able to put in new proteins in the cell. You want to maybe delete some genes and therefore delete some protein products from the cell. So, all of these require certain manipulations of the cell line. So, here in this picture on the left hand side, you can see the picture of jellyfish, and what you will notice is that the jellyfish appear greenish, and this jellyfish actually glows in the dark. So, they are present in the sea, in environments where very little light penetrates inside, and yet they have this ability to glow in the dark. And so, scientists isolated proteins that helped this jellyfish to glow and they called them the green fluorescent protein. And the green fluorescent protein has become a very important tool in biological sciences, which has helped us in characterizing localization studies of proteins of our interest. And how does this work? So, for this, let us look at this cartoonic representation. Suppose we have a number of these organisms here, and we consider each of these organisms to be different proteins present in the cell. And so, if you are to take a snapshot of the cell, which is for a cartoonic representation is a maze. So, if you look at the maze or the cell from the top, then you see these different proteins, but you do not know which protein you are looking at because all of them would appear the same. But if you are able to put a tag on one of these proteins, and if you are able to make that protein fluoresce, that is, it is able to emit a certain wavelength of light, and therefore you can see that protein, that light, then you can track that protein in the maze, and therefore you can track that protein in the cell, right? So then how do you add this GFP in a protein, and how does this GFP function? So for that, let us come to some basic understanding of recombinant DNA technology. So as we know, DNA is a double helical structure, right? And one of these strands of the DNA would be used to produce the messenger RNA, which would be coding for your proteins, correct? Now, suppose this is the region of the DNA that would code for the messenger RNA and therefore the protein. And if you are able to then isolate out just that sequence of DNA that strictly corresponds to the protein. So, this is the DNA that strictly corresponds to the protein that we would like to produce, and if we can insert in the front or at the back DNA that codes for GFP, then the whole thing would be transcribed as a single mRNA, which would be GFP with, say, the protein of our interest, right? And if you have GFP at the back, then it would be GFP here. And when the protein is produced, the protein would be folded conformationally, and when the protein is produced, it will also have this little GFP. If you have tagged it in the front, then it would be in the front. If you have tagged it at the back, then it will be at the back. So, this would be the N terminus of the protein, the C terminus of the protein. So, N is for the amino terminus and C is for the carboxy terminus. So, carboxy terminus. So, you have the GFP. Let us look at this once again. So, this is the gene that corresponds to the protein of our interest, right? Now, if we strictly take out just the DNA that codes for this protein and we fuse it with the DNA that codes for GFP, you will be able to produce one single protein that has GFP attached to it. So, this is the GFP, this is our protein. Okay. So, what would the GFP do? So, as I said, GFP would fluoresce. What is fluorescence? So, fluorescence is nothing but, so if there is the protein, when it is fluorescing, it would be excited by a certain wavelength of light. So, suppose this is the maxima of excitation, lambda max of excitation. Okay. At this wavelength of light or in this region, if a light is shown on the GFP molecule in this region, then the GFP molecule, the protein, the chromophore within this protein goes into an excited state, and then it emits light of a larger wavelength or a greater wavelength. So, this is lambda emission max. This is excitation max, this is emission max. So, this would be the emission. Okay. And you are able to see this emission because the emission is in a visible range. It is in the visible range. So, would be greater than 450 nanometers. In case of GFP, it would be more than 500, 510 nanometers, something like that. So, you would see this, you will be able to visualize this. You can see it with your eyes. You can also therefore capture images on the microscope. You can capture images under a microscope. All right. So, let us go back here. So, how do you then add GFP to the protein of your interest? So, you do this by recombinant DNA technology, right? You take the DNA that corresponds to the protein of your interest, you put that into a plasmid or a vector that you can put back into the cell. But when you do this, you also attach the GFP tag with it. It's not just GFP, there are multiple such fluorescent proteins that have been produced, all variants of the initial green fluorescent protein that was obtained from the jellyfish, and therefore these can be attached to the protein of your interest. Now, if you have a protein that is tagged with a green fluorescent protein and you have another protein that is tagged with red fluorescent protein, then you can simultaneously see in the same cell the localization of both of these proteins, one tagged in green and the other tagged in red. Okay, so here you can see an image of a cell with the mitochondria. So, mitochondria, as I had explained in the first lecture, they are the organelles that are responsible for providing an energy. They are the energetic source of the cell. That is a very simplified way of saying it. They are also very key components of multiple metabolic pathways. So, these mitochondria here are stained in red, and you can see the protein of interest has been colored with GFP. So, it has been tagged with GFP, and therefore, because these proteins can somewhat localize in the mitochondria, you can see some mitochondria which also show this green dot. Okay. So, thus it is a very useful tool to visualize proteins in living cells. But how do you put this DNA in the living cell? Because you have artificially created this piece of DNA, right, with the GFP attached to it. Now, how do you put this into a cell? So, there are various mechanisms by which you can insert the DNA of your choice that you have created by recombinant DNA technology into mammalian cells, and we are going to look at some of these methods in greater detail. So, the first and almost a very efficient and very easy methodology to do is by the means of viruses. Okay. So, let us understand this in a little detail. So, what are viruses known to do? Viruses are known to infect cells. And what do we mean by infect cells? What they do is they transfer their genetic material into cells. Cells they infect. So, this genetic material may be DNA, or RNA. Okay. So that is what viruses do. Now, this is a very simplified look at a virus. So suppose this is a virus. In this case, what I am trying to depict is more of a bacteriophage or a virus that infects a bacteria. So within this virus is the genetic material, which in this case is a DNA. And when this phage infects a bacterium, it injects this genetic material into the DNA, into the bacteria. So the DNA is injected into the bacteria. This is the virus, where the outer part of the virus is basically proteins and inner core contains the DNA, and this is the bacteria. So what happens to the DNA once it enters the bacteria? The DNA then starts, the DNA is then the virus DNA is then transcribed to produce virus RNAs, including virus mRNA. Okay, and therefore you can produce virus specific proteins. So the same situation applies to mammalian cells. So in mammalian cells you can have viruses, enveloped viruses which have a layer of lipid around them, they have proteins, and they also have this genetic material in the core. The genetic material may be DNA, may be RNA, okay. And in the cell, when this infects the cell, the viruses enter the cell, then the whole structure of the virus is disassembled. The genetic material is released and the genetic material enters nucleus. So if it is DNA, it could directly be integrated, it could integrate into host chromosomes or host DNA. If it is RNA, then it has to be reverse transcribed to produce DNA, and then that can be integrated into the host DNA. Once integrated into the host DNA, host DNA would then help to produce virus specific RNA, including virus mRNA, which can produce virus specific proteins. So given this understanding, if we can take the virus genetic material and into that, if we can cut it out and insert the DNA of our choice, DNA of choice, which may be a DNA that codes for protein, maybe say insulin, or maybe say some other protein, maybe an immunoglobulin. If you can put it into this DNA, that is the genetic material of virus, then we can form these virus particles, these different virus particles with the genetic material being this, with a DNA of our interest in it, right? And therefore, when these infect the cells, they would also be transmitting this DNA into the cells, and therefore, what we end up is with, if this infects cells, what we will end up with is a situation where cells produce mRNA and therefore protein that we have introduced into the viral DNA. Okay, so now given this understanding, we can now understand how viruses can be used to can be used as agents that can so given this understanding, we can now imagine how viruses can be used to manipulate cells. So viruses can be easily used to introduce proteins into cells. They can also be used to introduce interfering RNAs into the cells, so that you can decrease the expression of proteins. So viruses in that sense are very versatile tools, and the method by which you introduce DNA of your choice into cells by using viruses is called transduction. The second methodology is microinjection. So what is a microinjection? Now we all understand what injection is, because I think we have all grown up with a fear of the needle, and it reminds us of the stressful times we have spent in front of the doctor holding a syringe to our face. But microinjection is also something very similar. So you have a needle, and if you have DNA in a solution, you can use this needle to insert the DNA in the solution into the cells. But as you can imagine, the cells are really tiny. They have a diameter of roughly about 10 to 15 microns, and given the size, the needle that would be inserting DNA into these cells would also have a very small bore, which would mean that the size of the needles would also be really small to be able to insert DNA into these tiny cells. So in this picture, you can see a needle here on the figure A. You can see this is the needle, and the needle already contains this solution with the DNA. And in the figure B, you have an egg cell, and the egg cell is being held in place with a suction force of a pipette, and therefore the cell wouldn't be able to move. So you can hold that cell, and now you can slowly inject the genetic material into the nucleus of the cell using this needle, and thus you can achieve microinjection. Now, as you can appreciate, microinjection therefore would have an efficiency which is almost 100%, because you have efficiently delivered the DNA into the cell. However, the drawback of this method is that you can not do it for a very large number of cells, because as you can imagine, this is a manual process. It requires trained hands, it requires and it requires to be done one cell at a time, and because of this, this cannot be achieved in bulk. But with a virus, you can achieve transfer of DNA into cells of your interest in bulk, and viruses are also known to achieve this at high efficiency, even in cells that are otherwise difficult to be manipulated. So, the third method that we are going to talk about is called lipofection. So, what is lipofection? Now, the word itself may give you some hint, because there is the word lipo. Lipo comes from lipids. So, the cell membrane is basically lipids and proteins, and the lipids actually orient themselves so that they form a bilayer, and this is because the lipids have a hydrophilic head and a hydrophobic tail, right. So, let us go back to this. So, the lipids would have a hydrophilic head, a hydrophobic tail, and in the typical plasma membrane, they orient themselves to produce a bilayer of this kind. This would be really tightly packed and would produce a bilayer like this. Now, if you can produce lipids, if you add lipids into say water, what would happen? Now, if you put a drop of oil on water, what happens? The oil floats, right? The oil floats on water. And also, if you try to mix this oil and water, would it mix? It would not, right? It would not, because the oil is highly hydrophobic and therefore would like to always separate itself out. So, these lipids, if you take the lipids and if you try to put them in water, they would also try to separate out, and what they do is they form some kind of vesicles, some kind of droplet like structures. So, in this image on the right, you can see these droplet like structures. These are again, they form these bilayered membranous structures as depicted here. On the left, the cartoonic depiction, you can see the bilayered structure, and on the right you can see how these droplets actually look under a transmission electron microscope. Okay. Now, if you can mix DNA with these lipid structures, then the DNA would form a complex within these vesicles or within these droplets. And because the droplets can be easily taken up by the cells, the cell membrane also has these polar head and hydrophobic tail containing lipids. So, they can easily be taken up by the cells, and therefore, this is a good, efficient, quick method of transferring DNA into cells. So, the advantage of this method is that it is quick, it can be done in bulk, and it does not have to use a virus. Sometimes using viruses in human cells would require using viruses that would otherwise be infecting us. So, if you do not want to go into all those protocols, all those procedures, you can directly use lipofection to introduce the DNA into the cells. Okay, so the fourth method is called electroporation. So, again, the word itself would give you some hints as to what it is. So, electroporation is nothing but where you are using some kind of electrical changes, electrical charge to cause changes in the membrane. So, such changes would help the cells to take up the DNA. So, what is generally done is that for electroporation, there are something called electroporation. You have a cuvette, and the cuvette is nothing but a structure with these metallic plates on either side, which can then be put under an electric field. So, if you put your cells plus DNA into this cuvette, and if you provide the requisite voltage, so the things that we regulate are voltage, resistance, and so on. If you can provide the requisite voltage and current, then you will be able to make transient changes. So, what we make is transient changes in cell membrane. And so, these transient changes help in the uptake of the DNA. So, the transient changes help in the changes in help in uptake of DNA by creating short lived or transient again pores in the cell membrane. All right, so that was electroporation. Now, the last method that we are going to talk about is actually calcium phosphate DNA co-precipitation method, and it is very widely used. It's a very cheap method to do, easily can be used for bulk samples, but however not all cell types can be very efficiently manipulated by this method. So, what is done is there is a co-precipitation of the DNA of your choice with calcium phosphate, and this co-precipitates then attach to the cell. So, you take these co-precipitates, you put it onto the cell and they attach to the cell membrane and they are taken up into the cell through endocytosis. So, this method is also an efficient method, can be used for in bulk for a large amount of cells, and for the cells in which this method works, it is really highly efficient. However, as I mentioned, not all cell types can be manipulated by calcium phosphate method. All right, so let us move on to the next part of the lecture, where we are going to talk about some applications of animal cell culture. Now, before we go into applications of animal cell culture, let us recapitulate what we had learned in the very first lecture. So, in the very first lecture, when we were discussing about why we need animal cell culture, we had seen that there are a lot of different viruses which infect us, which infect humans, but wouldn't infect necessarily a bacterial cell, or even for that matter wouldn't infect mouse. So, in order to study those viruses, in order to study how those viruses infect human cells, how those viruses replicate within the human cells, how are they then produced in large numbers within the human cells, and how can you stop their production in the human cells, you need to grow human cells, right. So, viruses which can infect human cells wouldn't infect bacteria. Then again, if we are infected with a virus, then as an immune response, our body produces something called antibodies, and these antibodies would recognize that virus and will clear it out of our system. So, nowadays when we hear about coronavirus, we also hear about antibodies that are produced in infected individuals and are there in circulation in the serum, in the blood of the individual, right. So, these antibodies, when we would like to study these antibodies or how are these antibodies produced, we again have to look at the cells, the human cells that produce these antibodies. In this case, these cells are called B cells. So, the B cells are the cells in our body that produce these antibodies. So, in order to study that, we have to do animal cell culture. Then again, there are proteins in the human body that require something called glycosylation. And to study glycosylation, we again need to study the proteins produced in animal cells or produced in human cells. For example, if we again look at this immunoglobulin, right, the antibody. So, the antibodies are also glycosylated, which means that certain sugar molecules are added to this protein chain, and this glycosylation occurs within the human cell. It starts in the endoplasmic reticulum, it matures in the Golgi, and finally the protein or the immunoglobulin or the antibody is exported out of the B cell, or it is present on the surface of these B cells. So, these are the various reasons we discussed as to why we need animal cell culture. Now, we are going to look at certain applications of animal cell culture, and the first one listed in your syllabus is erythropoietin. So, what is erythropoietin? Now, erythro is something that relates to your red blood cells, and poietin is would mean that it has the propensity to produce these red blood cells. So, something that acts as a growth factor to help in the production of red blood cells, that is erythropoietin. So, erythropoietin is a protein. It is encoded by our DNA, but it is not produced in all cells of our body. Very few cells and some specialized cells in the liver and the kidney are known to produce erythropoietin. Okay. And erythropoietin is also a glycoprotein, which means that again these sugar moieties need to be attached to the protein chain to produce this functional, fully functional and active erythropoietin. So, how is the production of red blood cells regulated by erythropoietin? So, as I mentioned, erythropoietin would be produced by your kidney and liver cells, and this is produced in response to the number of red blood cells that are in circulation. So, red blood cells are produced majorly in the bone marrow. So, when you have very little red blood cells circulating in the blood, as is the case for patients who suffer from anemia, then the kidneys are instructed to produce more erythropoietin. So, greater amount of erythropoietin would reach the bones and the bone marrow, and therefore, the bone marrow would start to produce more red blood cells. So, this is like a feedback mechanism. So, low number of red blood cells in circulation would induce production of greater amounts of these erythropoietin, the growth hormone, which would mean that greater amounts of erythropoietin will reach the bone marrow and therefore would increase the production of red blood cells. Now, once the red blood cell production has increased, then there would be a large number of red blood cells in circulation, and therefore the kidneys would now understand that there is large number of red blood cells in the circulation, and therefore they do not need to produce so much of erythropoietin. So, erythropoietin production would decrease, and therefore little erythropoietin would reach the bone marrow, and thus the whole synthesis and production of red blood cells is controlled. Now, if due to some reasons a patient requires red blood cells in the body, it might be due to anemia, or it might be due to one of these diseases such as cancers or chronic renal failure, then it is sometimes advisable to give erythropoietin to the patient, so that you are not artificially injecting blood into the patient, but rather helping the body of the patient to produce more of these red blood cells. So, then erythropoietin, how do you produce this erythropoietin? You can produce erythropoietin by using a combination of recombinant DNA technology as well as animal cell culture. So, using recombinant DNA technology, you will be able to produce these DNA that would code for erythropoietin, the protein, and if you insert this DNA into a proper cell, which could be a human cell line or similar kind of eukaryotic cell that is able to properly fold and glycosylate erythropoietin, then you can also produce that protein in those cells. So, commercially, erythropoietin is produced in CHO cells, which are Chinese hamster ovary cells, and erythropoietin is, so this is a very good example to see how microbeads are actually used to produce proteins, recombinant proteins in industry. So, if you can recall, we had spoken about spinner flasks. So, spinner flasks are used to produce or generate human cells or animal cells in bulk, right. But spinner flasks are mostly designed for suspension culture cells, so cells that would not require an adherent surface. But you can use these spinner flasks even for adherent cells, and the way you can do it is by using something called microbeads. So, if you have these microbeads, you can grow the cells on the surface of the microbeads, and therefore, you can grow a large number of cells. So, CHO cells are very well adapted for this combination of microcarrier beads and spinner flasks, and so the CHO cells are grown under these conditions to produce erythropoietin. So, the other prominent biopharmaceutical product that requires animal cell culture is the production of clotting factors. Now, before we go into what are the clotting factors that are produced in the biopharmaceutical industry using animal cell culture, let us have a quick view of the clotting factors themselves. Okay. So, we understand, like it is a very basic understanding, that if we have a cut, then after some time that cut would heal, and the way it would heal is by blood clotting. So, how does this process take place? So, this is a very regulated process, and it is a multi-protein pathway, signaling pathway process, by which some proteins which are called proteases. And what are proteases? So, trypsin is a protease. Remember, we had spoken about trypsin as an agent that would help you to detach cells from culture. And trypsin is also present in our digestive juice. So, it is a protease, meaning it is able to break down protein. So, the clotting factors, many of them are actually proteases. They help in converting clotting factors which are inactive into active clotting factors. And why is this essential? Because in normal conditions in our body, when we do not have a cut or a requirement for these clotting factors to be active, you do not want clots to be formed. And if clots are formed, it can lead to very severe conditions such as stroke. So, stroke is nothing but the formation of these blood clots in the microvasculatures. Alright. So, because of that, we need clotting factors, and the clotting factors have to be regulated. Multiple clotting factors are proteases, and they help to convert inactive clotting factors into active clotting factors. So, similarly, here you can see on the left hand side it's kind of a flow chart to depict what happens, what are the key steps in formation of a blood clot. So, tissue factors and other factors such as factor 7A, they all combine and help to convert factor 10 into 10A, and this process is regulated by a combination of two other critical clotting factors. These are factor 8 and factor 9. So, factor 8 is actually a cofactor for factor 9, and factor 9 is a protease. It is also a protease, which means that it will be able to convert the protein on which it acts into an active factor. So, factor 9 is a protease, and it is itself present in an inactive conformation or in an inactive form until unless it is cleaved to produce 9A. So, factor 8 with factor 9A converts factor 10 into 10A, and the entire process helps in conversion of prothrombin to thrombin, and thrombin then finally acts on fibrins, and it converts these soluble fibrins into insoluble clots. So, this is basically in a gist how clotting factors work. Now, absence of any one of these clotting factors can be very lethal, and in fact a number of genetic diseases are known to be caused by the it's very rare, but it's known to be caused by the absence of these clotting factors. For example, factor 8, and the absence of factor 8 is known to cause a disease called hemophilia A. So, hemophilia A is the disease caused by the genetic absence of the clotting factor 8. Clotting factor 8 is again a glycoprotein, and as I have already explained, it is a cofactor for factor 9. And clotting factor 8 is involved in the production of factor 10A with the help of factor 9A. And clotting factor 8, again similar to erythropoietin, is produced in the biopharmaceutical industry through CHO cells. However, in recent times HEK, which is human embryonic kidney epithelial cells, so HEK cells have also been used to produce clotting factor 8 in the biopharmaceutical industry. Then we come to the next clotting factor, which is clotting factor 9. And clotting factor 9, the absence of clotting factor 9 is known to cause the disease hemophilia B. This is again a genetic disease, and this clotting factor 9 is also a glycoprotein, but it is a protease. Now, what I did mention before is that clotting factor 8 is produced by liver cells. So, special cells in the liver called the sinusoid cells, they produce this factor 8, as well as monocytes present in blood produce factor 8. Factor 9 is also produced in the liver, and commercially factor 9 is produced in CHO and HEK cells. Now, to come to some fun facts. So, the European royal families are known to harbor the mutations in factor 9, and therefore are known to have members of the royal family affected by the disease hemophilia A. And Queen Victoria is supposed to be the first person who had this mutation, and her children and grandchildren transmitted this mutation through the various royal families of Europe, and therefore this hemophilia is known to or is also called the royal disease. Alright, so when there is a cut, you need a blood clot to prevent bleeding. But after the formation of the blood clot and after the wound has healed, you need to remove that, and that is also essential. So, for that there are other proteins that help in dissolution of the clot. So, tissue plasminogen activator is such a factor which is very helpful in, or which is absolutely required for the dissolution of the clot. And tissue plasminogen activator is produced by endothelial cells, the vascular endothelial cells. Now, tissue plasminogen activator, also called TPA, it acts by activating plasminogen into plasmin, and this plasmin actually helps in the dissolution of the clot. Commercially, again, just like factor 8, factor 9, as well as erythropoietin, TPA is also produced in CHO cells. Alright, finally we come to another important therapeutic agent called monoclonal antibodies. Alright students, so far we were looking at clotting factors, we were looking at growth factors. Now let us look at antibodies. Now antibodies have become very important in therapeutic aspects. They are used for therapy against cancer, they are used for therapy against viruses. So let us look at how antibodies are produced in the biopharmaceutical industry. So here on the left you can see a mouse that is being injected with virus. So what happens when you inject the mouse with the virus? The body of the mouse would recognize the virus and would start producing something called antibodies. These are proteins, also glycoproteins. These are proteins that would be targeting the virus and helping in the destruction and removal of the virus from the body of this mice. And as we have discussed before, the cells that would produce these antibodies are called B cells, right. So here is a pictorial representation of a virus, and as you can see, the virus on the surface, it can have different proteins, which here has been depicted with different colors. So each of these proteins can be targeted by different antibodies, right. So this protein is targeted by this antibody, and so each of these antibodies are actually produced by different B cells. So this brings us to the concept of epitope. As I explained to you that this virus will have multiple proteins on the surface, and each of this protein can be recognized by a different antibody, and each of these antibodies are actually produced by different B cells. Again, even on a single protein, you can have different parts of the protein recognized by different antibodies. Therefore, an epitope would be a part of the protein or a protein that would be recognized by an antibody, right. So, when a virus infects us or infects say a mouse, then different antibodies are produced against that virus, because on the surface of the virus you can have different proteins, and even in a single protein you can have different parts of the protein recognized by different viruses. So, in our body, when antibodies are produced against viruses, it is always polyclonal, which means different cells, different clones of B cells are producing these antibodies. Whereas for therapeutic purposes, it may be necessary to produce monoclonal antibodies. Why? Because we may require to target a very specific part of a protein, say a part that has been mutated or that is different in a patient, but under normal circumstances that part of the protein wouldn't be present. So you would want to target antibodies against just that part of the protein. So how do you actually produce these monoclonal antibodies? So the whole field was sort of revolutionized by two people, Cesar Milstein and Georgios Koller, and they also won the Nobel Prize for this hybridoma technology. So the ingenious method that they developed was to combine two cell types. So you have a you have a myeloma cell, which is a cell of the B cell lineage, which is able to produce antibodies. So you have a cell that produces antibodies, right? And because it is a cancerous cell type, therefore it is immortal. Again, you have a B cell which produces antibodies that you would like to produce in bulk, but because it is a B cell, it is a normal cell, therefore it has a finite replicative capacity and it cannot be grown in culture for an indefinite period of time. But you want this B cell which is producing this antibody which targets a specific epitope of your interest. You want to keep growing this B cell so that you keep getting these antibodies. So you need to prolong the life of the B cell. What they did was to combine this B cell with this myeloma cell, and therefore the resultant cell type has the capacity to produce this particular antibody that you wish to produce, as well as is immortal. So, if you look here, these myeloma cells are immortal, and there is also something else written here. It is thymidine kinase negative. What this means is that this cell type is not able to synthesize DNA by itself. So, this being thymidine kinase negative is not able to initiate what is called a de novo DNA synthesis, and therefore would require some intermediates to synthesize DNA. Whereas, if you combine it with the B cell, because the B cell is not thymidine kinase negative, therefore, the resultant cell would be able to synthesize DNA. Now, because this myeloma cell is thymidine kinase negative, it is not able to grow in this specialized media called HAT media. HAT stands for hypoxanthine aminopterin thymidine. So, in this HAT media, the myeloma cells are not able to grow because they are TK negative. So, they will not be able to use these intermediates to produce their own DNA. However, these B cells are TK positive, and therefore, they can grow and survive in the HAT media. So, this is a selection criteria to ensure that your myeloma cells do not survive in the end, because they will not be able to grow in the HAT media. Your B cells alone will not be able to survive very long because they have a finite replicative capacity. So, they are not immortal. But if you combine these two, these cells are immortal, they will be able to produce the antibody of your choice, and they will be able to grow in the HAT media, right. So, using this, they were able to produce something called hybridoma, because it is a hybrid of these two cell types. So, using hybridoma technology, a lot of antibodies have been commercially produced and raised. So, one example here is a monoclonal OKT3 antibody, and this antibody targets the CD3 protein on the surface of T cells. So, T cells are also involved in immune response. So, we have B cells that produce antibodies, and T cells can directly attach to cells that have invaded our body and can kill them. So, these T cells have a protein called CD3 on the cell surface, as depicted here on the right. So, in this cartoonic depiction, you can see CD3, and the OKT3 monoclonal antibody attaches to the CD3, and this causes the T cells to finally go into something called apoptosis or programmed cell death. So, why would you require such an antibody? Why would you want to kill your T cells? Because your T cells are there to protect you. So, you would require to suppress immunity when a patient is being transplanted with an organ. So, in order to avoid that organ being rejected by the patient's body, by the patient's own immune system, you would require to suppress that immune system, and this monoclonal antibody OKT3 is used in such patients to avoid organ rejection. Now, the second example of such a monoclonal antibody is Herceptin, and Herceptin is a monoclonal antibody that binds to the HER2 protein. Now, you may have already heard about HER2 protein, because HER2 protein is often mentioned in the context of breast cancers and ovarian cancer, and HER2 protein is highly overexpressed. Overexpressed meaning if the HER2 level is in normal cells is very low or minimum, but in cases of these cancers, the HER2 expression exceeds the normal expression levels. Therefore, it is an overexpression. So, HER2 is highly overexpressed in some types of these, in some subsets of these cancers, and therefore, to treat these cancers, this Herceptin monoclonal antibody is used, and the monoclonal antibody, what it does is it binds to this HER2 protein and it causes multiple effects including internalization of HER2, it blocks dimerization, therefore it blocks the different effects of the HER2 receptor. Alright students, that is all for today, and in the next lecture we are going to see how animal cell culture can be taken to the next level in using tissue engineering and stem cell technology. Thank you.